Hey, we are starting to level off on people logging in, so I'm going to get started today. Thank you for joining for another session of OMF Journal Club. Today we're going to talk through this recently published paper from our Montreal Center, also in collaboration with our Melbourne Center, that's titled Haptoglobin Phenotypes Stratify Post-Exertional Cognitive Dysfunction Associated with Altered Cerebral Oxygenation and Metabolic Signatures in Long COVID. So, for those of you that have joined Journal Club before, you'll recognize this housekeeping item slide, and apologies for having to repeat myself on this, but just a reminder, if you could please post your questions in the designated Q&A channel. I will try to answer things as I go along if I see them, but I'll also get to questions at the end of the session today. To give some context for the perspective that I am bringing, I am a researcher, not a medical doctor. So, I am generally cautious about the claims that we try to make based on the data that we have available. But I will do my best to extend beyond just talking about the simple results and draw some connections to other research and talk about potential implications for people with long COVID as well as ME/CFS. But I do have to make it clear that anything that I say in that realm still needs to be thoroughly tested. And as I am not a medical doctor and, I don't get to have time to talk through personal details, I can't provide medical advice. I am going to walk through this as my personal way of explaining things to hopefully give you some better insight into what this paper is really talking about. But if there's something that still doesn't make sense, the way I've phrased it, please feel free to throw something in the Q&A, and I will try to reframe things as best I can so they're understandable. I do also want to mention quickly for the ME/CFS audience, I know the paper is looking at long COVID, but there is direct relevance to ME/CFS here as well, and I'll talk about it in more detail later. But this work is sort of a companion project to one that we recently published in ME/CFS specifically, and it does help to provide a hypothesis for how ME/CFS and long COVID might be biologically connected. So there is some direct relevance to the ME/CFS population here as well. All right. So I'm going to start out just trying to give some context that is useful as we're walking through the paper, and then I'll go through the results, the figures, and then get to our broader implications for the field at large. So this abstract is just the high-level summary of what this paper is looking at. So the problem that they're addressing here is that clinically we see heterogeneity in the cognitive function of people with long COVID. I'm sure that's not really surprising to anybody listening today. But the objective here is to really investigate the role of the haptoglobin phenotype in that heterogeneity of cognitive dysfunction. So this study specifically had 44 people with long COVID and 20 short course COVID controls. So these were people who had COVID but recovered quickly within 2 weeks. And so that's considered a control group that we're comparing the long COVID group to. And the participants provided blood samples, did cognitive testing, and had brain oxygen levels measured before and after a passive stress test, which in this case was a mechanical stimulation protocol using a cuff on the arm. And the study also tracked trajectories over time. So they went through 3 follow-up study visits that were separated by, I think it was 3 months' time in between each of those. So there is a longitudinal component to this study as well. And we'll get to the main findings on the next slide, but I have here just a quote from the abstract that gives sort of our high-level relevance. These findings support haptoglobin phenotypes as promising candidate biomarkers for biological stratification in long COVID, pending validation in larger independent cohorts. So, what are the actual key takeaways from this paper? Overall, what it's showing is that there are some clinically distinct subgroups to the long COVID cohort. So, compared with haptoglobin 1 individuals, and I'll go through what that means in a minute, haptoglobin 2 allele carriers with long COVID exhibited greater fatigue, poorer physical function, and more severe post-exertional symptoms. And so some of the specific findings that feed into that overall main conclusion are that haptoglobin phenotype is independently associated with physical function, fatigue, and post-exertional symptom severity in long COVID. The haptoglobin phenotype is associated with both cognitive response to exertion and cognitive resilience. Haptoglobin 1-1 specifically is associated with more efficient oxygen extraction under exertional stress in long COVID. And the haptoglobin phenotype is associated with distinct metabolic responses to stress. Now, I know that's a lot of detailed information right off the bat, but I'm going to point to each of these key findings as we walk through all of the results. So hopefully this will all come back around and make more sense toward the end of the webinar today. But I wanted to put that out up front so that you know kind of where we're going with this paper. And just some other key terms that I want to cover before we get into the results that will hopefully help us all, be on the same page with what we're talking about today. Haptoglobin is going to be a word that comes out of my mouth a lot during this session. So what is haptoglobin? It's also abbreviated HP for these purposes. It's a protein that is produced by your liver, and its main job is to bind to hemoglobin. So that's the oxygen carrying protein in red blood cells. And when red blood cells break down, it releases the hemoglobin, and the purpose of haptoglobin is really to clear it from the bloodstream before it can cause damage. So it plays a role in controlling oxidative stress and inflammation more broadly. And there are different types of haptoglobin that people can inherit. And so there are different structural versions of the protein that are called phenotypes. So I'm going to go briefly through what each of those phenotypes are. So haptoglobin exists in 3 structurally distinct versions. So we mentioned on the last slide, there's haptoglobin 1-1. There's also haptoglobin 2-1 and haptoglobin 2-2. So which one you have depends on which gene variants you inherit from your parents. So if both of your parents have the haptoglobin 1 variant, you are haptoglobin 1-1. If you got one of each, you're haptoglobin 2-1. And if your parents gave you the haptoglobin 2 variant, you're haptoglobin 2-2. So in general, HP haptoglobin 1-1 is the smallest structure, and HP 2-2 is the largest structure. And that difference in the structure of the haptoglobin changes how well it works. So haptoglobin 2-2 is associated with reduced antioxidant capacity and is generally less efficient at clearing hemoglobin from circulation compared to haptoglobin 1-1. And I think we'll see kind of the result of that through this paper. But under physiological stress like exertion, that difference kind of gets amplified. And I think that is really coming through strongly in this population. The other term I want to cover just really briefly is cerebral fractional tissue oxygen extraction, or cFTOE, I'm going to call it. It's a measure of how efficiently your brain is pulling oxygen out of the blood flowing through it and actually using that oxygen. You measure it generally using non-invasive near-infrared spectroscopy. So there are some sensors that are placed on the head that kind of measure how blood oxygen changes in the brain tissue underneath. So in general, a higher cFTOE means more efficient oxygen utilization. Okay, so those are just the brief terms that I wanted to cover that will hopefully help explain some of the context for this paper. And I want to walk through the results now. And just a reminder, I'll bring up the key takeaways from the figures as we're going along. But of course, if something that I say doesn't make sense, again, feel free to add a question to the Q&A and I'll try to spot it and reframe things as we go. So first up is our typical table of clinical and demographic characteristics for the study participants. So I won't go through this in too much detail, just noting that there is a long COVID column, so that is designated LC, and there are 44 people in that column. And then the short course COVID controls are designated SC, so that's the right-hand column, and there are 20 of those. And the main things I'll just mention, there was no significant difference between the average age and BMI for the cohorts. And as you would expect, the long COVID cohort had a higher symptom burden across all the different questionnaires that were used. So the next thing is just actually looking at the distribution of the haptoglobin phenotypes in both of the cohorts. So this is kind of the prevalence of each of the phenotypes in long COVID in the center and the short course COVID cohorts on the right. And you can see a pretty similar distribution where close to half of the people have the haptoglobin 2-1 phenotype and a quarter-ish each for haptoglobin 1-1 and haptoglobin 2-2 phenotypes. So really, I think the important thing for this table is just when we mention, differences in outcomes for the different phenotypes, this is, still a significant portion of each cohort, depending for any of the different phenotypes we're talking about. It's not just a handful of people. It does affect quite a few. All right, so kind of the meat and potatoes we're getting into now. These are bar graphs that are looking at symptom severity based on different haptoglobin phenotypes. So let me just pull up my quick little laser pointer here. So just looking at panel A, for example, along the x-axis, we have the haptoglobin phenotype. So HP11 is in blue, HP21 is in purple, and HP22 is in red. And then along the y-axis, you have, in this case, the SF36 physical functionality questionnaire. But going across, we have two different questions from the DSQ PEM questionnaire, the physical fatigue portion of the MFI-20, and then the reduced activity portion of the MFI-20. All right. So on the very left panel here, we have where the haptoglobin 2-1 and the haptoglobin 2-2 groups in purple and red have lower scores, meaning worse physical function compared to the haptoglobin 1-1 group. The next two, like I mentioned, are looking at specific questions of the DSQ PEM survey. So panel B is looking at, in quotes, dead, heavy feeling after starting to exercise. Panel C is looking at feeling physically drained or sick after mild activity. So in both of those cases, you can see a similar pattern where the HP21 and the HP22 phenotypes in purple and red experience these symptoms more severely than the HP11 phenotype. Then panels D and E, again, are components of the MFI-20, measuring fatigue. And again, you see the same kind of general pattern where HP22 is showing the greatest impairment. Now, one thing I want to mention that isn't pictured, but the team also ran a regression analysis to understand if age, sex, BMI, or disease duration impacted these results at all. And they found that the haptoglobin phenotype was independently predicting physical function and fatigue severity, even after adjusting for those variables. So those conventional clinical variables didn't contribute significantly, and the haptoglobin phenotype alone explained a large proportion of the symptom severity variation that they saw. So the key takeaway that I'm going to point to again here is that haptoglobin phenotype is independently associated with physical function, fatigue, and post-exertional symptom severity in long COVID. And I will note for the majority of these figures, they're just showing the results for the long COVID cohort because there weren't statistically significant results in the short course COVID cohorts. Those are generally in supplementary figures. I'm just not showing them, but they are there if you're curious to look at it. Okay, so now we turn to the cognitive focus of the study. So cognitive function was assessed using the BrainCheck platform. And just to orient you a little bit, higher scores generally indicate better cognitive function. So on the left panel here, these are bar graphs again, and we're looking at cognitive function before, so T0 in blue. So this one would be at time 0 before the stress test, and then the red would be time 90, which is after the stress test. And it's again split by the haptoglobin phenotypes. So what you'll notice is that the only group that showed a significant decrease in cognitive decline immediately after the stress test was this haptoglobin 2-2 phenotype. So you see the statistically significant drop between the blue and the red in this right-hand group that you don't see with these other two phenotypes. So the next two graphs are looking at longitudinal follow-ups. So again, they went to, in the blue, visit 1, visit 2, and visit 3 that were all separated by, I believe, about 3 months. So 3 follow-up visits rather than just immediately after the stress test. And so in panel B, what you can see is that the haptoglobin 1-1 group, so this grouping of 3 over here, actually had a statistically significant improvement in the cognitive function in that visit 2 follow-up, whereas the haptoglobin 2-1 or 2-2 had relatively flat trajectories over time. And panel C is a specific component of the cognitive testing that measures delayed recognition. And you see essentially the same pattern here as you do with the overall cognitive function, where there's that statistically significant increase in the haptoglobin 1-1 that you just don't see with the haptoglobin 2-1 or 2-2. So this figure is looking at both the cognitive response to exertion in this panel A, but then also resilience, so like the ability to bounce back over time. And before I bring up our key takeaway, we have another view of kind of the resilience over time on the next slide. So this is a table that summarizes the trajectory of cognitive function over time compared to the person's baseline score. So you can see it's broken up by those that deteriorated compared to the baseline score, those that improved compared to the baseline score, and those that were stable compared to their baseline score. So there were 10 people in total that over that longitudinal 3-month time period between visit 1 and 2 showed a decline compared to their baseline cognitive testing. But what I think is notable here is that of those that deteriorated, only 1 had the haptoglobin 1-1 phenotype, whereas there were 6 and 3 with the haptoglobin 2-1 or 2-2 phenotypes. So combined with the last slide, what we're pointing to here is that the haptoglobin phenotype is associated with both cognitive response to exertion and cognitive resilience. All right. So now, given that, association between the phenotype and cognitive response and resilience, they wanted to explore a little bit more about what was happening biologically that might contribute to that. So this is looking at whether there's a connection to the cerebral oxygen extraction. So that's that cFTOE thing that I mentioned in the key terms area. So these are box and whisker plots. So just to quickly orient you, this kind of box in the center is showing the middle 50% of values in that group. And the line in the middle is the median or the midpoint of all of the values. And then the whiskers extend up and down to capture the broader range of all of the values. And then again, as pretty much every figure here, we have the haptoglobin phenotypes across the x-axis. And then the cerebral fractional tissue oxygen extraction, cFTOE number, is along the y-axis. And again, just a reminder, that's a measure of how efficiently your brain is pulling oxygen out of the blood, throwing through it, and using that oxygen. So I will note this is specifically reporting on the right hemisphere of the brain, so that's that little _R there. They noted that the left hemisphere showed similar trends, but they just weren't statistically significant, so they're only picturing the right hemisphere in this particular figure. So this panel on the left is at time 0, so that is before the stress test. And then this is at time 90, so that is after the stress test. And you might notice that the haptoglobin 1-1 phenotype, so that's this green box here and here, shows a generally higher cFTOE than the other two phenotypes, and that it gets essentially more pronounced after the stress test. So just a couple things to note from the discussion of the paper for extra context. The team also measured total cerebral hemoglobin, so essentially kind of the total blood volume in the brain, and it was fairly stable across all of the phenotypes and time points. So that is something that would suggest that the differences we're seeing aren't because these haptoglobin 2-2 patients have less blood reaching their brain than the others, but rather we're actually seeing differences in how efficiently the brain is extracting and using the oxygen from the blood. And then again, the short course COVID controls aren't pictured here, but they did, note in supplementary information, the phenotype differences in the cFTOE were not present with the short course COVID controls. So there were no significant differences across the phenotypes. And so again, that's kind of telling us that these brain oxygenation differences might actually be specific to the long COVID disease state and not just whether somebody has these haptoglobin phenotypes alone. So the key takeaway that I'm going to pop up from here is that haptoglobin 1-1 is associated with more efficient oxygen extraction under exertional stress in long COVID. All right. So then they wanted to take a look at things that are going on in the bloodstream using the plasma samples that were taken. And they did a metabolomics panel on those samples. And, based on quality criteria, they were able to analyze 26 specific metabolites. And all 26 of those are listed in this table here. And there are two main columns. So one is this p-value column, which is another statistical method for showing significance of results, essentially. And FDR is an additional kind of correction for running multiple tests on a lot of data. So this is actually really the column that we pay attention to in this case. And the arrows that I have along the left side are pointing to the ones that are showing statistical significance after that correction. So they were the ones that were different across the haptoglobin phenotypes at the post-stress test time point. So this is looking at a few of those metabolites in more detail. So panel A is isothianate, which is related to sulfur metabolism. Panel B is citric acid, which is a key intermediate in the TCA cycle, which is the main pathway that cells use to generate energy and taking place in the mitochondria. And then glucosamine here is a natural compound that's found in cartilage and plays a role also in mitochondrial function. So there's some biological relevance to these three metabolites that they chose to look at in more detail. So panels A through C are just the levels of each of those metabolites based on the haptoglobin phenotype after the stress test. And all three of them were lower in the group with the haptoglobin 2-2 phenotype, which is shown in red, and generally also lower in the haptoglobin 2-1 phenotype that's in purple. Now, panel D on the right here shows citric acid specifically before and after the stress test. So the left bar for each phenotype is the before the stress test, and the gray bar on the right is after the stress test. And this is showing that the haptoglobin 2-2 phenotype on the right here is the only group that shows a significant drop in metabolite level in response to the exertion. So then, given all of these different levels, they then wanted to look at how these metabolites correlated with cognitive function as well. So that's what the E through G panels along the bottom are. And what you can generally take away from this is that all three metabolites correlated positively with brain check scores. So as you move to the right on the x-axis, you also generally move up the y-axis. So higher levels of the metabolite as you go right mean higher scores on the cognitive function as you move up the axis. And they're just a little nuance to point out. This one is looking at isethionate with a specific portion of cognitive function. That's the delayed recognition. Citric acid was correlated with overall cognitive function scores, and glucosamine was correlated with the immediate recognition as opposed to delayed recognition. So again, something to note that's not pictured. None of these metabolite changes, again, occurred in the short course COVID controls. So the key takeaway from this figure is that the haptoglobin phenotype is associated with distinct metabolic responses to stress as well. Okay. Now I'm going to go through and reiterate all of those key takeaways and kind of what they collectively suggest for us before we talk about the bigger picture and implications for the clinic. So looking at these specific ones that I pulled up for each of the figures, just to remind you, the haptoglobin phenotype is independently associated with physical function, fatigue, and post-exertional symptom severity in long COVID. So again, it wasn't dependent on age, sex, BMI, or disease duration. The haptoglobin phenotype is associated with both cognitive response to exertion and cognitive resilience. So that was looking at, the difference in response immediately after the stress test and then longitudinally. The haptoglobin 1-1 phenotype is associated with more efficient oxygen extraction under exertional stress in long COVID. So that was looking at those cFTOE values being higher and that difference being more pronounced after the stress test. And then haptoglobin phenotype is associated with distinct metabolic responses to stress, which we just saw in the last main slide, where you had the different levels of isethionate, citric acid, and glucosamine. And then altogether, what that's telling us is that compared with haptoglobin 1-1 individuals, people with long COVID that had haptoglobin 2 alleles, so that's haptoglobin 2-1 or haptoglobin 2-2, generally had greater fatigue, poorer physical function, and more severe post-exertional symptoms. Okay, so then looking at the bigger picture, there are some details in the discussion section of the paper actually, but these are just a few areas that I want to highlight for you. So first, the obvious question, connections to ME/CFS. So I mentioned at the very, very beginning that this was kind of a companion study to one that we had recently published on ME/CFS. So this is the second paper this team has published on haptoglobin phenotyping. And the first was actually in people with ME/CFS, where they found the same pattern that haptoglobin 2 carriers were faring worse overall in these, post-exertional symptoms and symptom severity. And so that was actually kind of the rationale for studying it in long COVID, was because they saw it with ME/CFS first. But an important distinction that I want to call out is that the biology was a little bit different between ME/CFS and long COVID. So in ME/CFS, the team found structural changes in the haptoglobin protein itself and evidence of hemolysis. Whereas in the long COVID cohort, the haptoglobin structure was actually intact and the hemolysis wasn't present. So the authors of the paper are interpreting this as, or posing a hypothesis that this is kind of indicating that long COVID is potentially an earlier stage along the same trajectory toward ME/CFS, where the body's like compensatory systems, so systems that are responding to stress, are still a little bit better at doing their job, but beginning to strain. And that further down the straining pathway is kind of what we're seeing with ME/CFS. So again, that's just a hypothesis based on what they're seeing comparing these two cohorts. But I think it's, a compelling framework for maybe how these diseases relate to each other. So really would be interesting to test that further. A couple other things I want to point out is there are some findings and implications that I mentioned throughout this paper that are consistent with other things that are found in ME/CFS and long COVID research. So we mentioned that citric acid and glucosamine are connected to mitochondrial dysfunction. And I'm sure many of you know that mitochondrial dysfunction is a huge component of what we think is going on with ME/CFS. And I also talked a little bit about how haptoglobin 2,2, based on its structure, is generally less efficient at clearing hemoglobin, and that can be connected to oxidative stress. Oxidative stress is a finding that, our group at Stanford and others have pointed to in ME/CFS and long COVID. So there are some consistencies across research studies that I think are really nice to see. And then the other connection I want to make is actually with a project that we have going on right now that is our large-scale biomarker project called BioQUEST. And this is actually an example of how we're carrying this work forward. So this paper is, contributing a hypothesis that we're essentially able to test as part of this BioQuest study. So it's in a larger and more diverse cohort. We're going to be linking haptoglobin phenotyping with, multi-omics and clinical data and all this other testing that we're doing within BioQuest, and importantly, having both ME/CFS and long COVID cohorts within the same study, so we can hopefully, maybe see validation of these results, but also potentially answer some more of the questions about how ME/CFS and long COVID are connected in this particular sense. All right, I'm pulling up therapeutic implications here. Again, I have to restate that I am not a medical doctor. This is, extending beyond just the simple results of the paper. So anything that I say in this realm needs to be tested, and please don't change anything about your treatment strategy without talking to a physician. So the first portion I want to mention just in terms of therapeutic implications is that haptoglobin phenotyping seems to be a promising method of stratifying studies. So you could use it to reduce heterogeneity in whatever patient population you're including in a trial, actually trying to identify patients that might be at higher risk of post-exertional cognitive impairment and making sure that you can, account for that correctly. And then also you could consider targeting specific portions of the population for specific interventions based off of, this phenotyping that's done. And I put up two different versions of therapeutic targets that, might be a logical conclusion from some of these results. We mentioned citric acid and the citric acid cycle in general, having issues in ME/CFS, and this is an active area of exploration in the field, maybe most notably with oxaloacetate trials. Again, that's still in development and needs to be handled very carefully, but there is a connection to ongoing treatment research there. And glucosamine is actually something that can be supplemented, and we saw that at lower levels for the haptoglobin 2,2 phenotype in response to the stress test. I will note that there are different forms of glucosamine supplements that do different things. So that is obviously really important to tease out before we are able to say what's really going to be beneficial. But glucosamine is actually, supplementation with glucosamine is actually used in other conditions for pain relief as well. So there is just, another relevant connection to ME/CFS. Again, would need formal testing, but something I wanted to point out for you all. Okay, that is the main bits of the paper. And as I get to the questions, I do just want to actually share something with you all about one avenue through which we are trying to make sure our research findings also reach physicians. We want to make sure that we have a pipeline to actually using research results in the clinic. And so I just wanted to note for you all that we do actually have a channel on a platform that's called ViewMedi, which is a resource that's specifically for physicians, where we can take published research like this and add it to the research section of our channel so that it does get surfaced as new information to physicians that might be relevant for how they are treating patients with ME/CFS and long COVID. Okay, so there were some questions that I wanted to get through that were submitted ahead of time, and then some I think that are coming through in the Q&A. So let's see. There is a question. There are a few bits to this question, so I'm just going to read it in full. Sorry for a little bit of length here. So that given that SARS-CoV-2 infection can trigger ME/CFS, did the study assess whether any long COVID participants developed an ME/CFS phenotype after infection? Beyond requiring persistent symptoms for more than 3 months, what additional screening was used to characterize the cohort? Were participants assessed to determine whether their symptoms reflected an IACC phenotype, or could the cohort include a broad range of persistent post-COVID symptoms like anosmia, skin symptoms, or other non-ME/CFS conditions? And then final piece of this was, if ME/CFS phenotype was not assessed, how can we determine whether the haptoglobin findings reflect long COVID broadly or a subgroup with post-COVID ME/CFS? Okay, I'm going to try to keep this a concise answer that I think addresses all parts of this. They were assessed for post-exertional malaise. So that makes it much more likely that the people with long COVID involved in the study match ME/CFS criteria rather than long COVID more broadly. The BioQUEST study I mentioned may help us determine whether the findings are consistent for the more broad definition of long COVID, or whether it is more specific to the ME/CFS phenotype. But I would expect specifically the results from this paper to be more on the ME/CFS phenotype side of long COVID. Somebody asked, is this something that patients can test in a commercial lab, or is this only available in research settings? So right now, this is largely available in a research setting when it comes to the haptoglobin phenotyping specifically. You might be able to get the genotype information from genetic testing, so you would be able to know kind of the HP11, HP21, or HP22 information. You wouldn't necessarily know the structural integrity of the protein from that. So wouldn't have that same in-depth information about the structure, but you might still be able to get the genotype from genetic testing. Let's see. Okay, somebody asked, so that haptoglobin 2,2 profile tolerates physiological stress very poorly. Does this genetic phenotype promote an accumulation of free radicals that causes a blockage of the Krebs cycle? Yeah, so I mentioned a little bit that the haptoglobin 2,2 phenotype is generally associated with less efficient clearing of hemoglobin, and that can raise oxidative stress. So that can impact the Krebs cycle, which is also known as the TCA cycle or citric acid cycle. It has a lot of names, but we saw some of those negative impacts, I think, with the citric acid levels after exertion. So, we're seeing some of those results coming through with this paper specifically. Let's see. How does a patient with non-existent haptoglobin fare? Are they worse off than those with haptoglobin 2,2, or does the body compensate in other ways? That is a very interesting question. I will say I don't definitively know the answer to that. I would have to look it up. I would imagine that there have to be some compensation avenues that the body has. I don't know what they are offhand though. So I don't know how to compare whether they would be worse off than haptoglobin 2,2 or not. Sorry for that one. Can you share again the platform for clinicians where you share research? Yeah, so it's called ViewMedi. It's VUMEDI. And it is unfortunately something that only physicians can access. So, we can't give access to patients as well, but we will hopefully soon be able to share just, a list of the things that we have on our ViewMedi channel if patients are interested in seeing what is being shared through that platform. But that is something that clinicians, physicians can access for free anywhere around the world. All right. I think I have gotten through the questions. Apologies if I missed anything. I'll leave it open for another minute or so in case there are last minute ones that come through. But thank you so much for coming to Journal Club today. Thank you for using your spoons on this and engaging with me. I really appreciate the thoughtful questions and your interest in our research. Someone asked, how could one evaluate different types of glucosamine? That would be something that you'd have to certainly do that in conjunction with a physician who is familiar with what your situation would be, as well as what each of the different types are doing. So with anything that has these different kind of formulations, there should be information available to physicians that details the mechanism of action or what's known about that particular drug or supplement in this case. So that would be something that would be good to consult with somebody who would be able to follow you through, each of those different things, or at least be able to identify what might be more likely to help in your circumstance. So unfortunately, I don't have a straightforward answer to that, but yeah, it would definitely be looking into what the different formulations do differently and what would be relevant for you in particular. What would be a potential benefit of knowing one's phenotype? Yes. So I think this is a great question that kind of leads to the concept of precision medicine. So, one thing, for example, let's say that you know that you have the haptoglobin 2 phenotype, and maybe when it comes to some kind of exertion, I would imagine you might consider supplementing something that would enhance your citric acid cycle so that the drop that you see with the citric acid after exertion, you'd compensate for that and maybe have better mitochondrial function following exertion. That would just be one, hypothesis of how that would carry through to a clinical knowledge, but knowing what the different phenotypes do in response to exertion in this case might help kind of determine different treatments or things to try for your specific case that you might not bother with if you have a different phenotype. Okay, I think that's all the questions then. Again, thank you so much for joining and for engaging with me. I really appreciate it, and I hope you all have as good of days as possible.